The work that I'm going to talk about actually was done by a whole bunch of other people, as usual, and I just want to call them out. Mostly work by Tony Rodriguez and Eli Bowen, and a bunch of funding, which we're very grateful for. Neural nets today, deep learning, for instance, does two things really well. It classifies and does big data and does games, which are big data applications. And things that animals, including humans, do are classifying and one-trial learning and attending and tracking and searching and sequencing and changing sizes of things and relations among things, following rules, localizing, navigating, focusing, deciding, ignoring, inferring, and of course for humans, especially language. The hypothesis among deep learning is that we keep climbing the deep learning tree and we'll get to the moon. The other possibility is that no amount of climbing will get us there, that there are additional things to learn. So these are the things that are missing that I just said. By contrast, there are circuits in the brain that convey a highly constrained but very wide, what you might call instruction set, if you will, of elemental operations. And these span, of course, they're brain circuits. They span from raw perception to abstract concepts. And it's possible that we may be able to mine these for additional algorithms. And quite pragmatically, that's what we're going to talk about, is understanding brain circuits as circuits and mining them for algorithms. In the process, some deceptively simple capacities may become better explained, and one of the examples that I'm going to highlight at in the last segment of the talk is human judgments of similarity. That is, when are two things similar to each other? When do two things look alike? I hope to argue that these algorithms are pointing towards suggestive, novel, unusual way, easily understandable, nothing mystical about any of these, but unusual, non-standard approaches to understanding these things that is yielding fruit. Little outline. I'm going to talk about brain circuits and what constitutes them. I'm going to turn those brain circuits into hierarchies of circuits that I'm going to formalize with formal treatments in terms of mathematical grammars. Not linguistic grammars per se, just formal grammars. Then I'm just going to jump to a section here where I'm going to talk about how we use these. Examples of why they're pragmatic, what kinds of algorithms come from them, what kinds of applications can we do, and I'll highlight two examples. One is learning relational structure, another is a novel set of algorithms, family of algorithms for supervised learning without any error correction whatsoever. Which is weird, and as you'll see, is efficacious and much less expensive than standard approaches, which is perhaps of interest. Then, having jumped to these applications, I'm going to jump again to two separate segments. One is a inquiry into what happens as brains grow within the evolutionary constraints of allometry, and what kinds of changes might be occurring as we go from small mammalian brains to large mammalian brains. And finally, I'll jump again to this question of when do two things look alike. So a lot of jumping. What I'm going to do is give you a really fast, if you will, flyover of a bunch of topics, but I'm going to try and dig into each one of them enough so that they become intelligible as isolated incidents, and then try to bring them back together. So, start with brain circuits. We study brain circuits like everybody, many people do on 3 timescales. There's the construction of the brain circuits over evolutionary time, and we look at the constraints of that. There's integration of built-ins on one hand and experiential data on the other hand over developmental time. And there's the acquisition of facts and procedures, which is learning on the third hand, which is the thing that, of course, most gets focused on. The mammalian brain arose arguably 200 million odd years ago, and invented the neocortex, but the neocortex only operates in conjunction with and in tight loops, feed-forward and feedback loops, with other brain structures. So we're going to take a few steps through some of those brain structures. And I'm going to just flash by a few of the constraints that we try to take seriously. So there are excitatory and inhibitory cells. Almost all of the transmitter in the brain is glutamate. Almost all of the receptors in the brain are glutamate receptors, which is very interesting. And it's not that you can't do without the rest of these. But the sheer numbers of glutamatergic circuits may be illustrative. These kinds of circuits are probabilistic. They're slow, they're sparse, they're low precision. Those are the engineering characteristics, if you will, of these circuits. And that's nasty, because this is a kind of circuit component that you would not want to build stuff out of. In fact, no self-respecting engineer would ever use these components. And the trick question is, the principle, that's it, the principle is to try to take seriously some of these constraints, and I'm going to give you several of them, and see how it is that we can build high-precision components from low-precision constituents. And of course, this is something that's done in engineering quite a bit. It's not novel to us at all, but it's intriguing to see how it arises in some of these cases. So this is one example of a design constraint. Another is the thing I mentioned, that the cortex works in tight loops with hippocampus and the amygdala and the striatal complex. That's a constraint. And each of those very distinct circuit designs for thalamocortical loops, which we'll talk about, striatal complex, hippocampal structures, and the fields of the amygdala, are the very different circuit designs of those are also design constraints. It's not a bunch of neurons in a network, neuron soup, if you will, but rather a bunch of very structured circuits that are, each of which are doing different things and conferring different capabilities to the overall interacting circuit. These are embedded in a big architecture of posterior and anterior cortex with corticostriatal loops and corticolimbic loops. And over evolutionary time, as small brains go to big brains, that's a design constraint, but as small brains go to big brains, these things take over the structures of the brain, such that this cortical, striatal, palatal, thalamic loop is repeated over and over again. There's one for vision, there's one for audition, there's one for touch, and there are ones for things that we don't have behavioral types of terms for, but they are if you will, the architecture of mammalian brain, and they are the thing that we're striving to understand. That's a design constraint. And as they get larger, the anterior cortex gets disproportionately, allometrically larger, and that makes some of these connection pathways disproportionately much larger, which very much changes the behavior of these things, certainly in certain mental illnesses, but also in normal everyday behavior, and that also is a design constraint. So let's take these samples of sampling of constraints and see what kinds of things we can turn them into. This will be a series of cartoons on just thalamocortical loops. So here's a very simplified cartoon of thalamocortical loops, and sensory inputs, for instance, come in from to the core or specific matrix, if you will. They go up here, they go up to superficial layers, they go down to both layer 5 and layer 6. They feed back in these different fashions, depending on whether they're feeding back to the specific or non-specific thalamic regions. And so what we're going to do is cartoon our way through a couple of derivations of what different components of these loops are doing in our hands when we simulate them. So these two pictures here are now even further simplified. The two pictures on the left and right are the same. All I've done is highlight the core portion, if you will, of the thalamocortical loop and the matrix portion of the thalamocortical loop. They're the same structures. I'm just highlighting parts of them because I'm going to do two separate treatments of them for a moment here, running through the core loop and making a set of hypotheses that in our hands, what this thing is trying to tell us it does, in simulation, and then turning those into algorithms, and then what this thing seems to do, and then put them together. Yeah, question? Yep.
What do you mean by the matrix?
I only mean that this part of thalamus, so there are parts of thalamus that are called core thalamus and parts of thalamus that are called matrix thalamus. It's just a naming scheme that is a pretty standard scheme for those guys. So we're just borrowing their terminology. It's two different parts of the thalamus that are differentially connected. So the core thalamus provides inputs, takes inputs from the periphery, and provides inputs to cortex. The matrix thalamus gets no extrinsic inputs whatsoever. The only inputs to it are from cortex. They're feedback. And it then feeds again to a loop to cortex. And so it's doing something presumably quite different. And we'll look at that. So core and matrix loops, I'm going to treat them separately and then bring them together. So core loop first. We're going to think of the core loop in terms of static images. That is, you're staring at something. You're looking at a person or a slide or a flower or a face. And you're fixating on it. It's staying still. And of course, you're learning a bunch of these. So you've got a whole bunch of flowers. You've got this kind of flower and that kind of flower, etc. Okay, so for each one of these, it comes into here, goes up to middle layer cells, goes up superficially to superficial layer cells, and these in many people, in our hands and in many, many labs' hands, are excitatory inhibitory loops that do something like lateral inhibition, which behaves like competition. A bunch of excitatory cells get the same input or similar inputs, and they talk to their local inhibitory cell, and the inhibitory cell, once it's triggered by responses from one of these responds, talks to the inhibitory cell turns everybody in the neighborhood off, including the guy that turned the inhibitory cell on. So that lateral inhibition is causing a race. It's saying some of these superficial cells will win that timing race because they'll get to spiking first, they'll activate the inhibitory cell, and they'll turn off everybody. And indeed, this is the kind of sparse behavior that you see in superficial layer cortical cells all the time, that they respond and then they stop responding because you've got rampant inhibition. So we think of that input as kind of clustering, because as these inputs continue, as you get all these different flowers, a vector, a spatial pattern that comes in, each time one of these comes in that's similar enough, that's sharing enough of its vector, it's close enough in the input space, it's differentially strengthening the parts of the input, the parts of the vectors that are shared. So the parts of these flowers that are similar to each other all become learned, they become differentially strengthened in these synapses, and then the inhibition stops it. And that's the feed forward piece of this. These then feed down. I'm ignoring layer 5 for the moment because we're going to come to it in the matrix piece. So they feed down to layer 6, which feeds back to, in part, this inhibitory layer, the nucleus reticularis, which inhibits the input, topographically, inhibits part of the input. What's it inhibiting? What it's inhibiting is whatever was activated by these guys. What was that? Whatever was activated by these guys. What's that? It's the set of shared inputs. It's the category of the things that are similar across these flowers. That's being inhibited from the input. The input's still there. It's static. And so the input comes in again, the flower comes up, but now parts of it are inhibited. What parts? The shared parts across all these different flowers. So now what's going to come in is the difference between the leftover, the remainder of the shared parts. Once you subtract out the shared parts of the flower, what is the different parts here and the different parts here and the different parts here? Those now become input. Those get learned, and they feed back and inhibit. And what you get is an algorithm for hierarchical clustering. What it's basically doing is saying, oh, I've got a flower. Oh, I've got a red flower with large petals. Oh, I've got a it doesn't matter if it's this particular hierarchy, and of course it's not meant to be any specific cortical representation. What it's meant to do is just be a cartoon that illustrates that you get large categories, smaller subcategories, and smaller sub-subcategories. What's happening is the superficial cortical cells are iterating over time. You get a response. You subtract part of it with nucleus reticularis. You get a next response, which is this successively lower level, a description of the difference between the members of this big category. So you're getting category, subcategory, sub-subcategory, read out over time by the successive behavior of feed forward, feedback, feed forward, feedback. We strip away all the biology and we say, what kind of algorithm is that? It's doing hierarchical clustering. It's saying, oh, I'm finding a winner, I'm training the cells, and I'm subtracting the mean of the winners from the input and then iterating. And if you look at that as a hierarchical clustering algorithm, you find that it's actually a really, really good one. And hierarchical clustering turns out to be something that's very, very well studied. There are whole journals called hierarchical clustering. This algorithm that came from nothing but this simple simulation of these feed-forward and feedback circuits turns out to do a very good job head-to-head against standard hierarchical clustering algorithms. So we can look at this and we can say, by the way, recognition and categorization are actually the same thing. This is broad categorization. When you get down to recognition, you're just doing special case categorization. Recognition is just a special case. It's categorization with small categories. And notice we're getting that thing that I said, high precision computations from low precision components, because it's iteratively reusing them over time. One of its tricks. We can do the responsible computer guy thing and say, what are the space and time complexity costs? And it turns out that, again, especially if you take into account appropriate parallel architecture, these things are very efficient in terms of space, very efficient in terms of time, and in fact are performing at neck and neck with the standard, best industry standard hierarchical clustering algorithms, which is nice, but it's a little weird because it wasn't supposed to be a hierarchical clustering algorithm. It was a set of cortical feedback loops. That was the core loop. Let's do the matrix loop. And again, then I'm going to put these together. So here's the matrix loop. Now notice, it's going to take as its input whatever the superficial layer is building from it, from the core loop input, and it's going to feed that down to layer 5, which is then going to feed down to this thalamic structure, which is going to feed up to layer 1. And I'm going to call out these synapses just for a moment. This is just a scare slide to remind us to say there are lots of data suggesting that there are time-dependent, sequence-dependent synaptic learning algorithms. That is, you can find things that will respond, put simply, will respond very differently to AB, the sequence A then B, than they'll respond to B then A. And there's, again, big literature on it, and what we're doing is borrowing some of that, and so we're just going to make synapses up here that are taking these inputs and learning that. So we're going to use now, instead of static inputs like a picture that you're fixating on, time inputs. This says, what does it say? Bird. Bird. And what it comes in is as a sequence of, just like you see the still frames of a movie and you see them in rapid succession and it appears to you that things are moving. Same thing is happening with Audition. You're actually getting successive input, individual snapshots of inputs, and you're putting them together over time. And that sequence of left to right is what comes in. So here's what's going to happen. Spoiler. The input's going to come in. It's going to learn its categories, if you will, up here. These are going to feed back so that the second one is going to be arriving here as the first one, which already responded, is now feeding back, and they're going to converge up here. So the second one feedback at the same time as the first one comes in, and in fact that timing is set in this loop, so you get the feed forward, and you get a function that's a little bit like a hash function. In our hands it's actually a thing called a Bloom filter, and it hashes, so it's doing the opposite of clustering, the opposite of what these superficial cells are doing. It's saying, even for very similar inputs, I'm going to give them very different labels, very different addresses. That's the simple intuition for it. So it's going to give unique identifiers, if you will, patterns of firing, which are going to come up here, and that sequence is now going to be learned. The chain of this, then this, is going to be learned in these superficial synapses. So, in this case, the thing is trying to do sequence completion, and that's what we're recognizing is the ongoing attempt to complete this sequence. This also can be turned into an algorithm. It also can be analyzed. The interesting thing I'm just going to point out in that analysis is that there's always a question of capacity. That is, as you add in the x-axis here, as you add size to the network, you add synapses in this case, how many more data? How much more data can you learn? And the answer turns out to be both empirically in this network and in a little formal treatment that Rodriguez did, that these grow linearly. As you get bigger circuits, you're learning linearly more sequences. That's a very unusual characteristic of networks of this kind, as many of you know, and it's a very useful fact because instead of throwing lots and lots of computer at these things and hoping that they will get big enough to do big data, in this case, you can actually build larger and larger and larger ones of these, and they're not giving you diminishing returns. Okay, we've got the core loop doing clustering and categories. We've got the matrix loop doing sequences. And a quick side note, are there interactions between these? Well, that's a problem. When things are moving, presumably you're not getting the same input, you're not fixating on that flower. And so it's not going to, if you subtract you take that loop and you subtract that thing, are you going to get the subcategory of the thing? Not if it's moving. Well, interestingly enough, it leads to a suggestion that maybe when things are moving, you're not going to do that iterative, successive deepening category-subcategory thing, but rather you're going to just get category. Oh, and then the next time category, because these things are moving over time. We came across this thing that has no relation to our work, but it's an interesting illustration. These are moving dots that are changing. So they're changing their colors and fixate. No, I'm going to go back up. Okay, so fixate on the little white thing in the middle, this thing in the middle. Fixate on that. Watch them change. Fixate on the middle. Fixate on the middle. Fixate on the middle. And it looks like they stop changing, right? And of course they're not, so let me do it again. Now, don't fixate on the middle. Now just watch one of them. Watch it change. And you'll see that, sure enough, it keeps changing. It's just that when you fixate on the middle, you don't see that. So the motion, and so that's because it's not moving relative to you if you're fixating on it. The motion is maybe interfering with the iterative deepening that you would otherwise do. It's a hypothesis. This works in a bunch of different cases, and Alvarez has this great set of illusions that do this. So these ones are changing shape, and I broke it. Changing shape, and again, as they move, you fixate in the center, and it looks like they stop changing shape, but of course they're not. Okay, whoops. Okay, so there might be an interaction. Let's now look at the combination of these two things. You've got a category thing and a sequence thing. Before I do it, I'm just going to say we're going to put them together, but we're also going to put them together with and this is probably the smallest summary of decades of work that you'll see in 2 slides or 3 slides. Similar cartoons that I could give in a talk of modeling of the striatum, modeling of the hippocampus, and I'm going to call out this idea of match and mismatch that we think the hippocampus is doing, that many people think it's doing, and the amygdala, and take all those cartoons and embed them in an integrated architecture where we have inputs and we go successively downstream, and I'll just use this cartoon for convenience as we go forward. This gives us a little summary of, all in a couple of minutes, of algorithms that are derived from these circuits. The reason I'm jumping to this little summary is because now what we're going to do is take these individual algorithms from these components and put them together into interactions and see what they do. And this is where we're going to get to applications and a bunch of novel algorithms. So the individual constituent algorithms are clustering, categories, hierarchies. Thalamocortical circuits, again, sequences, as we saw, and chaining. Striatal complex in many people's hands, reinforcement learning, hippocampal fields, time dilation, and I want to actually, I don't have it here, but match and mismatch. That's what we're going to think of, loosely speaking, not that loosely speaking, of as the instruction set. What we're going to try and do is stay at that level. If that's an instruction set suggested by bottom-up models of individual circuits, then what larger instructions do you build from that instruction set? What procedures get built from that as you let these things interact? So here's the integrated operation over time, if you will. One area, the output of one area, doing sequences and categories. The outputs of one area are the inputs to the next area, naturally, and the outputs of those, the inputs to the next. So if this is doing a sequence, this is doing a category of sequences, and this is doing a sequence of categories of sequences, just because the outputs of this are the inputs to that, etc. So sequences of categories of sequences are formal grammars. They're simply, that's what grammars are. Linguistically, you can think of them as, a sentence goes to a noun phrase and a verb phrase, and then the noun phrase goes to an adjective and a noun. Again, notice, what's the sentence? It goes to a sequence, noun phrase, verb phrase. What does the noun do? It's a category. What's the noun phrase then expand to? A sequence, adjective, noun. Grammars are like that. Again, we don't think of them as linguistic grammars, they're just formal grammars, but we're going to give some treatments of these in a moment. So now we've got this instruction set, and it's building a set of shared data structures that are being passed around, nested sequences of categories or grammars. Okay, what we're going to do with this now is go after data. We're going to go after, and the data that we're going to focus on are virtually all, almost all moving things, things that change over time. And one of the reasons is that that's critical to most of our processing. So in this picture, you don't see much. You see like a little building over there. But if you watch the picture now, yeah, all of a sudden, there's that guy. So let me do it again. Yeah, that guy. You can't help yourself. That's what you see. And it's because motion does this to us. We've got these built-in, not built-in, it's, many, some are indeed built-in and some are learned templates that operate over time. And so I'm going to skip that. So we use these to do little learning, spatiotemporal units that are hierarchically building grammars, and they're being used to learn the little pieces of movies over time. So this is what they look like, hierarchical categorization of spatiotemporal receptive fields. Little things that move, like that, and we build them up, and notice, a thing to say is, time is not its own representation here, but rather, as you go successively downstream, you're getting a whole bunch of, just in cartoon, bunch of successive levels of these hierarchies, which look in cartoon like this, and they get more and more complex. They're little grammars, each one of them. And then, of course, one of them feeds back, so it's feeding back, and we can prune backwards so that we have a complete absence of explosion of the combination of these things, because all you store are the ones that actually occur. And once you get high enough, every time you take a step upwards in this hierarchy, you get many, many fewer things that actually occur in the world. Let's look at some quick examples, and this is just going to be a flyover. So learning relational structure. So we do things like training, looking at an object, but not statically. We look at it over time and space. And so here's an example of a car, and look at the car far away. Look at the car intermediate. Look at the car near. What do we get? We get a bunch of pictures of a car, and we know they're all the same thing because they're contiguous. And yet this one doesn't resemble this one at all. And yet we know that they must have the same structure, and we use that information. This is work largely by Eli Bowen and Carrie Jarrett and a number of other students who are trying to learn this structure and apply it across these time sequences, so that we can address it with questions of the kind that were, if it was further away, what would it look like? If I turned a little bit, or if it turned, what would I see? So now, even the beginning part of the truck doesn't resemble the back part of the truck, but because it's all contiguous and because it's learning these relations, it finds the invariant structure and tracks that thing over time. I'm calling that invariant structure small scale because we also do this large scale. That is, here we are moving through a big space, and if you watch right over here, it's expanding a map that it thinks it's building. It is building. As it moves through these spatial structures, so it can't see all of it at once at any point, not like the car where it was turning around, walking around the car. And it's going to build these things, and it's going to do things, among other things, like simultaneous localization and mapping, or SLAM. It's going to figure out when it's actually gotten back to a particular point. This is actually in a video game. We also do it by running drones, and this is just a moment for me to say these are all intrinsically parallel algorithms. They're not algorithms that have to be parallelized. They're born parallel, they start parallel, and so they cry out for a certain kind of hardware, and we have done some work on implementing that hardware, which I will not say one more word about, but we build these things into drones, and this one's a drone flying through the math building, and again, it's building a map as it flies. And here's one that's outside going around the campus. So skip over that. That's attempting to learn relational structure using these spatiotemporal hierarchies. Now I'm going to completely switch gears and talk a bit about the building of algorithms that are interactions among these constituent algorithms. So I talked about this little list. Let me pick out two of them, clustering on the one hand and match and mismatch on the other hand, and show an algorithm that was built by Ashok Chandrasekhar, in which it's got a clustering piece and a match-mismatch piece. Here's what it's going to do. Intuitively, it's simply going to say, I want to categorize things unsupervisedly. I'm going to take supervised data, so we're just artificially applying this to supervised data just to demonstrate a point, which is it's actually going to do extraordinarily well at supervised data, even though it was very clearly not built to do any such thing. It's going to take supervised data, do unsupervised classification of it, that is just by similarity, and then it's going to say when do the labels match and mismatch, and it's going to iteratively deepen a hierarchy. So here's a bunch of data, and it's going to say, okay, here's, imagine it's high-dimensional data, you've got red things and blue things and green things, and what an SVM or any deep learning system or any standard error correction system will do is it'll find a separator between one of these categories and the others. It'll find this very expensive to determine separator set of hyperplanes. What does this thing do? It says, now I just want to find categories of things that are similar. And then I'm going to say, oh, some of them are pure with respect to the labels after the fact. So I'm going to leave those alone, like this guy right here. I'm just going to leave it alone. And then I'm going to take the ones that are not pure with respect to the labels. This has red things and yellow things in it. And I'm going to say, oh, well, just do unsupervised categorization again, so that I'm going to get, okay, here's a nice pure one, pure. But here's an impure one. Okay, do it again. And just keep iteratively deepening only in the places where there are difficult discriminations, which is intuitively has an interesting relationship with what SVMs actually do. And so the outcome is very, very different. What it's going to do is building a generative hierarchy that actually reconstructs the data. It's showing you things like there are different kinds of stars. Here's a type of star over here, and here's a type of star over here, and they're different, which this thing cannot tell you. And it's able to reconstruct the data. So that, there's a difference between, of course, discriminative algorithms and generative algorithms. The popular wisdom is that generative algorithms are richer and more expensive. This is richer and not more expensive. So here, first of all, we'll just show that it performs equally to or better than SVMs. So here's work by Eli Bowen on showing that the CSL algorithm is it has a better ROC curve than the SVM on a bunch of visual data. But the time cost, interestingly, here's the time to learn for SVMs, here's the time to learn for CSL. This is just a parameter, the branching factor, just so that we had a parameter to vary, and it doesn't. It barely changes it. All of it is like an order of magnitude cheaper to train. So I'm going to argue that it's equaling the performance of standard classifiers, in fact, outperforming them sometimes, costs significantly less. It's building these generative representations, and it emerged from something that wasn't meant to be a supervised algorithm at all. And I'm going to skip this entirely. I know, but we'll take questions. Skip that entirely and go to similarity. Each of those little spatiotemporal little units is constantly doing match-mismatch. It's saying, what things are similar to me? Well, similarity is not Euclidean. And when we do Euclidean distances between things, we're getting it wrong. If we do the Euclidean distances between these three vectors, for instance, they're all the same. The Euclidean distance or the Hamming distance in this case, they're all the same. But if we look at these pictures, this guy's over here and these guys are over here. These guys are closer to each other. So you might want some kind of perceptual distance that says that these guys are closer than these. What kind of perceptual distance? And by the way, assist from Captain Obvious. Here's another way to look at this. Here are two contrasts, NA prime, B and B prime, and the differences between them are exactly the same, but we see these as very different from each other, and these look much more similar to each other because of the contrast. What we want, and the distance measures will not tell us this. What you want is some distance measure where this distance is much less than this distance, right? Because you want to be able to register the perceptual fact, whatever humans are doing, that is saying, these guys look really much more different from each other. This is a big jump. This is, by contrast, by comparison, a smaller jump. And indeed, contrast is the thing to look at. So, look at the circuitry. I'm just going to whiz through this. Look at the circuitry. We've got feed forward and fan in and fan out projections that give us, in fact, typical receptive fields. Each one of these projection patterns is simply a matrix of the changes to this guy as a function of the changes to this guy. You can take all these mappings, these connectomes, little mini connectomes, if you will, and write them as Jacobians. And so, for instance, this particular one here is this Jacobian here. And we can use those, and what I'll show is, briefly, just very quickly, is that when you do that, you do the math of non-Euclidean geometry that will give you similarity measures that match perceptual measures. And I'll show a bunch of empirical studies that do this. So all we're going to do intuitively is we're going to take the feature space of the vectors, map it into perceptual space with a Jacobian that maps it, and bring it back to say what's the distance that we should have measured. So if the Jacobian mapping from feature space to the psychological to perceptual space is this little Jacobian, then a distance using those Jacobians is a tensor, a metric tensor that is this form, this Jacobian transform times itself. If I take a straw man where we don't have a bunch of crosstalk in these, if we just had this cell projecting to this cell projecting to this cell, and no crosstalk, in this straw man you'd get, indeed, Euclidean distances. But of course, that's not, and you've got the Pythagorean theorem as a side effect, but that's not what's actually happening. Of course, what's really happening always is you've got a bunch of crosstalk. That crosstalk all by itself is what gives you Jacobians with off diagonals. That's not Euclidean. It's actually not affine. The general case of this is Riemannian. There's several ways you can treat it. We're going to treat it as Riemannian space because it's a very nice general way to treat it, and we show with differential geometry what you can do with it. So we're going to treat this as a Riemannian transform, and what you see then is that the distances between these things do not follow the Pythagorean theorem and are going to accurately reflect human judgments of differences. How do we do that? There's a whole field. It's called image quality assessment. And one of the subfields of image quality assessment is take two pictures, ask humans, how degraded is this picture? How bad is this one compared to the one on the left? And it turns out there is no ground truth for this. It's simply whatever humans do. And we love that, of course, because that's where the mystery lies. And so there's a whole field out there of image quality assessment. And as you can imagine, there's a buck in this gig. The field tries to come up with algorithms that estimate or guess what human judgments are going to be. The standard in this, the one that's referred to over and over again in the industry literature, is SSIM, an algorithm called SSIM. And what they do is they show humans a bunch of these pictures and then degraded versions of these pictures, and they do this typically kind of graph where they show the human dissimilarity versus the algorithm dissimilarity. And here's the kind of graph you get. So with Euclidean distances, if you use a Euclidean algorithm for dissimilarity, then you get a pretty crappy R squared. If you use SSIM, which is what the industry does, you get a much, much improved one. And if you use this Riemannian geometric similarity, you get a much improved one. And again, it's obvious why you do, because when you're looking at those pictures, and looking at the difference between them, the difference between them, you're using the fact that they're spread from side to side and projection fan out and fan in, that is changing the way you're going to judge the pixels side by side. They're not just pixel to pixel, which is what Euclidean would do, and they're not just a little Gaussian blur, which is what SSIM would do. There's something more complicated. And in fact, what they do is differential geometry, at least if you buy the idea that purely predictively, these things, the differential geometry version gets a much better estimate of what humans actually do. So what do you do with this? Well, it's one thing to say you can now guess what humans are going to judge. You could turn this backwards and say, oh, well, what if we wanted to compress an image? And the two measures of these are the error, that is the distance between the original and compressed, as measured by human perceptual judgment of that error, just like we were talking about, and the entropy, that is, how big is the file that you compress it to. That's what JPEG does, and it does it by using a thing called a discrete cosine transform. And the problem with this is that each one of these little components that it tries to decompose the image into is supposed to be orthogonal. They are orthogonal in Euclidean space, but they're not orthogonal in perceptual space. And it's easy to show this if you just put different weightings on, for instance, this one and this one, which are very different. They can actually look very, very similar to our eyes, and that can be taken advantage of in a compression algorithm. So what we do is, and I'm going to we take a feature space. What JPEG does, it goes to a DCT space and it builds a new version of this, which you can then build the error, measure the error and entropy of. What we do is we do this succession of generating the distances between. Here, you're going to see a little movie of running down the quality spectrum and showing what the corresponding error and entropy are, and all you're seeing is that if we do it this way, that we can get better, down here, better error and entropy trade-offs than you would get with JPEG. And so I'm going to skip that and simply show some examples. This is a classic example from the JPEG literature in which the problem is that these two things interact. And they're not supposed to, but they do. And so if you see a JPEG of it, this is what you get, is a bunch of noise. If you see one of these RHGpeg things, it performs much better because it's using the relation between the parts of the image to change your estimate of what the distance between these things are. And then it's building a compression based on that estimate. And so here you see that the JPEG ROC versus the RPEG ROC. The RPEG one is outperforming it. There's a bunch of measures that we can do with this. It works on any image. And so we can do the same thing with JPEG and RPEG of faces. And again, they outperform. And the cost of doing this is exactly the same as JPEG, because we can we can usefully simply rewrite the JPEG set of transforms in terms of Jacobians, which is useful all by itself, and then just show side by side what the comparative costs are, and they are the same, which is nice. So, quick and dirty summary. We take the bottom-up connectivity and cell responses of the way that a target cell responds given changes to the changes to input cells. We do differential geometry transforms on these, and we find a new error measure for image quality, and we find a new method for doing JPEG, and they have the same costs of as the extant methods. I'm going to skip all this and simply say a take-home. Take-home. Current systems are a surprisingly modest subset of the algorithms that we might be looking at. That's how I started. Maybe that's why humans are still doing a lot better in most cases, other than search over trillions of data points. Other than that, we still outperform them by enormous leaps and bounds. Brain circuits offer an alternative. There's a bunch of, not an alternative, a supplement, if you will, a supplement, a bunch of elemental operations that you can build things from, and they're atypical in psychology, they're atypical in engineering, but they're very normal in terms of brain circuits. And some deceptively simple capacities, I hope I've shown, may become better explained, like human subjects' judgments of similarity, including demonstrable and patented improvements over current standard widely used methods like SSIM and JPEG. And that's the name of that tune. Thank you very much. Questions? Yep. Sorry, you.
Please use the mic for questions.
Good. Great.
Thank you very much for the talk. You were talking about how we can, so basically your second point here, that the brain is can be thought of as being composed of a subset of different circuits that compose an instruction set. Then when you were actually explaining all of that, you mapped everything onto well-known computer science notions, such as hashing and all sorts of other algorithms, which can later be used to do all sorts of things with. My question is, why did you need the brain to do all of that? Why couldn't you just use the concepts that are very well known in computer science?
Thank you, and I love that question. The way to have done this correctly is to simply give all the credit to me. That is, I thought up all this stuff. I took the standard methods from the computer science literature and, you know, my little book of algorithms, and I invented and, invented a bunch of new combinations, new ways of using these that are turning out to do things like outperform JPEG. I'll take that credit. That's absolutely fine. Of course, I didn't do that. The answer is, there's a lot of algorithms, and you don't know, and I don't know, and our students don't know, and our postdocs don't know which ones to use, in which combinations, for what applications, in what sequence. We don't know any of that. What these circuits did was they actually showed us. We didn't invent this stuff. We mined in the mine to see what they were trying to, you know, sit up like a dog and bark. Thank you, Wally. What they were trying to tell us their behavior was, and to us, it would have been beyond bizarre if these had turned into algorithms that nobody had ever heard of. I'm not sure I know anything of the sort. Algorithms are algorithms. We got them. The question is, which algorithms are good models or appropriate models of different circuits, and how do those combine and interact to give us the overall behavioral algorithms that are giving us these effects, behavioral effects, estimates of data from human behavior, and direct applications that can, go out in the world. That's the way I think about it.
I had a second question, if you don't mind. So when in the first part of your talk, when you were talking about the core and the matrix loops, what I didn't understand fully was, especially with the matrix loop, that it doesn't receive any peripheral input. Right. It just communicates back and forth with the cortex. My question was, why does the cortex need the matrix loop to learn sequences? We know that the hippocampus is very good at learning sequences, for example. And to my knowledge, it doesn't directly communicate with the thalamus for that purpose. So what's the role of the
Good.
thalamus?
No, these are
here?
terrific questions. I'm going to keep taking questions from you. First of all, we know, notice that was him, not me. We know that the hippocampus learns sequences. Of course we do. No, we don't know that stuff. And I don't know it and you don't know it. And what we have is a bunch of data that is consistent with the idea that the hippocampus is learning sequences, A. B, we've got a bunch of data that other structures are doing things that are related to sequence learning that are beyond what the hippocampus does. And we know that, for instance, hippocampal field CA3 can have long-lasting firing. We also know that medium spiny neurons in the striatum can have long-lasting firing. We also know that cells in anterior cortex can have long-lasting firing. Why? Why did it waste all that energy on duplications of effort? In this case, it's not me to blame. It's the big designer. We don't know why those things were designed the way they are. We don't know why certain things are conserved over evolutionary time in mammalian circuitry. So the short answer to your question is, in our hands, we see these things actually performing those behaviors, and we try to understand, how can that behavior be useful?
Sorry, yeah. I guess my question was, where's the data that shows that the thalamus is in fact involved in that loop?
Oh, my goodness. As
opposed to the cortex just doing all that.
Yeah. There's not as much of it as any of us would hope, but there really is data. I don't have a slide like that. Let me send me, anyone who wants to know stuff like this, send richard.granger@dartmouth.edu, and I will send you references. There are studies like this, and some of them have to do with just the sequential nature of those superficial synapses that I did show some references to the data, but some of them have data on what things like a bunch of the relatively well-studied non-specific regions of the thalamus do. There are studies like this. Yeah. Now, are they conclusive? No, they're certainly not. No, they're not. Are they consistent with what we've modeled? Yeah, they're consistent. And my guess, I should have hastened to say from the beginning, are these models right? No, they're almost certainly not. They're intended to take the things that we know from anatomical structure and physiological operation that we can measure, like this data, and trying to find out what we see when we simulate them and then simplify them into algorithms. What we're hoping is that they're pointing us in a right direction, that they're on a right track, and that as we keep looping back to do things like tests of behavior, and a bunch of tests that I didn't show of fMRI and other tests that we do of single units, that we're trying to see, we're trying to refine these things and get better at them. So no, we don't have that locked in, but yes, there is data.
So one of the big puzzles has to do with why we have a neocortex at all. And so in a very kind of interesting, intriguing way, I think your work bears upon that, except that then I think, the reptiles and turtles that don't have a neocortex, they are doing pattern recognition. They're able to learn. They are able to discriminate similar from non-similar things. So is it just a weaker algorithm, or is it much the same circuitry, only it looks different because it's clumpy, or what's the story?
There's more than one answer to that, and that's a fantastic question. Again, the one answer is that different circuits, like, for instance, especially avian circuits, which are really different. And yet there's an awful lot of bird people who do, not to denigrate, but anyway, bird people who are studying a bunch of behaviors that are, to them, shared, they want to claim, between avian and mammalian species, and we don't know. We just don't know. And if they say they know, you send them to me, and I'll have a little talk, because we don't know. Of course we don't. Another answer to this is: are we just lumping things together? We say, oh, they learn. Oh, they recognize patterns. Oh, they can tell when two things are similar. In cases where these things are carefully tested, we often find that there are subtle and important differences between the ways that a mammal will judge two things to be similar and a pigeon will judge two things to be similar. And the field often focuses on the similarity between these things because it's amazing that the pigeon can do anything. And that's very valuable. But it may also be very useful to ask just this question, which is, if those are different circuits, are they doing different things, or are they accomplishing the same computation with different algorithms? I don't know, and I don't think anyone knows. But one of the things to carefully study is the differences in the behavior, and they're not to just lump them and say, oh, they learn, oh, they do pattern recognition. What we're trying to do is say, isolate very specific behaviors where we can control all the components of it, like take two images and have judgments of how different they are. And if a pigeon did that and had to operate those sliders, which they quite perhaps can do, would they give the same answers? If so, then we'd have to explain how those avian structures, which we don't understand well, and which we had to completely rename all of 10 years ago, how those structures are carrying out that similarity measure as opposed to the way that these structures are. But at least we want to get the behavior very specific first and not simply lump it all together. That would be my real answer. Yeah.
Andrea.
So I was just wondering, you mentioned the amygdala's role in slight toggle, I think you said, et cetera. I wanted to know if you wanted to comment on that just a little more.
Thank you. Great. So what we've spent a lot less time on the amygdala than we have on almost anything else. Sorry, amygdala people. But what we, in our hands, when we do the kinds of analysis that we do, what we find is that it is switching on and switching off cortical areas. So, and it's doing this toggling of only allow these regions to respond to this input, and then different state-dependent emotion, if you will. Now only allow this one and this one and this one to respond, and not this one, and to the same input. So it's changing again, in our hands, cartoon hands. It's changing the selection of regions, thalamocortical whole regions that are allowed to respond to an input, and maybe that has something to do with, and we haven't done anything. Again, I can't emphasize enough. We've done very, very little work. But the suggestion might be that this might have something to say about state-dependent memory, and that it because it's telling you why you want to be in a certain state, and you're going to get a different answer depending on a different recognition, literally, as simple as that, depending on what state you're in. And maybe a little bit of the constituents of what kind of different answer you're going to have, what's going to be emphasized and what's going to be diminished, depending on what that amygdala state is. We've done very little, very little. That's why I use the term toggles, because they're toggling on and off different possible responders. Yeah, you're out of questions. That's right.
I don't know if anybody's out there in the world communicating with you about questions. No? Okay. We can all ask Rick more questions outside where there should be some goodies waiting for us. Let's thank Rick again.
Thank you.